Hello GCSE biologists, welcome to my video where I'm going to be covering the entire Module 2 GCSE content on organisation. And to help you along with your revision, you can also access the free revision flashcards, which you can get by clicking the link below, putting in your details, and we'll send them to you immediately. And they are, yes, absolutely free. Now, if that's not enough, you can also access my resources by clicking the link below, and you will have access through that to lots and lots of study notes. You will get videos, you'll get exam style questions. Again, all absolutely free to help with your consolidation of your notes, to help with your GCSEs in the summer. Anyhow, let's crack on with the video.
The principles of organisation. What are these? Living organisms are made at a very basic level from cells, but they build up into more and more complex levels, going from cells to tissues to organs to systems to organisms. So what we're saying here is that living organisms are made up of systems that are organised into increasing levels of complexity. We'll start with the basics. Cells. What are cells? Remember that cells are the basic units of life, and they are the basic unit of all living organisms. And we have lots of different specialized cells, like muscle cells, like nerve cells. But when cells work together that are similar in structure, and they also carry out a specific function together, we then call that level of organization a tissue. Typically, muscle tissue will contract to bring about movement, as a good example. Then we get the level called organ level, where different tissues carry out a common function, working together, like the stomach, for example, which is made up of muscular tissues, glandular tissues, epithelial tissues, all working together to help with the digestion of food that we take into our bodies. Organs will then be organised into systems which carry out a much broader function that's essential for our survival. The digestive system is something we're going to look at in this video, and this includes the stomach, the intestines, the liver, the pancreas as well, and that will be addressed in much more detail in a second. And then the organism stage or level is when we have this complete living organism where all these systems work together in unison, and then we have a really complex living thing. So, digestive system, this is where we're going to start looking at examples. What is the purpose of our digestive system? When we take in food, the food we take in is often in an insoluble form. It's way too big to be absorbed into our bloodstream. Therefore, our bodies have to break those large insoluble molecules down into small soluble ones that can pass into our bloodstream and be delivered to our cells to provide our cells with all the nutrients they need for their survival. Digestion comes in two forms. We have mechanical digestion, which is a physical type of digestion involving the kind of grinding and the crushing up and the mushing up of food, where the food gets made into smaller and smaller parts, but there's no chemical change to the food at this point. Chemical digestion, on the other hand, is when we get chemical changes, and this is typically carried out by enzymes. Why do we need to do this? We need to make sure that our large insoluble molecules of food that we eat become small soluble absorbable nutrients like glucose, like amino acids, that can then enter our bloodstream. There's always going to be food that we take into our bodies that we cannot absorb because we don't have the right enzymes to do that. They will never be small enough to pass across the lining of our gut and enter our bloodstream. But we need to still get rid of these. We don't want these sitting around in our bodies. This undigested waste has to be egested, which means we go to the toilet and we get rid of it when we go for a poo. What is it then in our diet that we need to digest? There are three main big food types that we need to digest, known as the polymers. Polymers, poly means many, made up of many repeated units. Carbohydrates, for example starch, is our big polymer that we need to break down into sugars like glucose to be absorbed. Proteins need to be broken down into amino acids to be absorbed. And lipids, which aren't really polymers to be honest, but we're just going to call them that for the GCSE level. Lipids, fats and oils that we ingest have to be broken down into glycerol and fatty acids to be absorbed. To do any of this breaking down or digestion, as it's technically known as, we do need chemicals. And you can see in the 3 diagrams that we have chemicals named here, which are enzymes. We are going to look at these in more detail in the video today. And these enzymes are essential if we are going to chemically break down food to make it soluble, to make it absorbable. We're going to do a quick overview though of all the organs involved, from the mouth right down to the anus, to see how they are used in the digestion of our food in a little bit more detail. Starting with our mouths. When we take food into our mouths, the first thing we do is we chew it. We also have that feeling of saliva, and both of those things are really important. The chewing, the mechanical digestion, the physical process, using the teeth grinds the food up. It makes big food particles into smaller food particles. It doesn't chemically change them, it just gives them a bigger surface area. On the other hand, the saliva that's coming in from our salivary glands contains an enzyme called amylase. That's a chemical which begins the breakdown of starch in our food in our mouths. Going back to those salivary glands. Now, salivary glands are found typically in the cheeks, and we also have one under our tongue, and they squirt saliva into our food as it's being chewed, so it mixes into the food with the action of the teeth at the same time, and that starts the chemical digestion, as I mentioned, of starch into sugars. Once that has happened in the mouth, we then swallow the food. We call it a food bolus at this point. This passes down our throat into a tube called the esophagus, sometimes more commonly known as the gullet. And this esophagus has smooth muscle in its walls. It's literally a soft tube with muscle in the walls, and the muscles undergo little contractions called peristalsis that pushes the food down towards the stomach. Digestion itself does not occur in the esophagus. It's purely a way of transporting food from the mouth into the stomach area. Once in the stomach, we get more mechanical, physical, and more chemical digestion happening. The mechanical part is the churning of the food. The stomach wall undergoes contractions. The stomach itself is a muscular bag, and the contractions of that wall help to churn the food even more and also mix it with chemicals which are coming from the lining of the stomach. The 2 main chemicals here are hydrochloric acid, which is a very strong acid. It has a pH of around 1 to 2. And we also have protease enzymes being secreted from the stomach lining here. Now, the hydrochloric acid and the protease enzymes work together really, because what the hydrochloric acid does is it provides the optimum pH for the protease enzyme to start the digestion of protein on our food into amino acids. And the hydrochloric acid also helps to kill ingested pathogens, i.e. things like bacteria that might have come into our bodies along with our food. Now, at the same time, the liver isn't directly involved with digestion. It doesn't do digestion itself, but it produces chemicals that help digestion to happen. We'll address this a little bit more further on in the video, but just for now, we're going to mention that the liver is an organ that produces bile, and bile is involved with fat digestion and also helping to neutralize stomach acid. As I said, we'll address that a little bit more in the video in a moment. The gallbladder is a tiny little pouch. You can just about make it out on the diagram at the side here. So it literally sits under the liver, and what the gallbladder does is it's a storage bag for the bile. So the liver makes the bile and then sends it through a duct into the gallbladder, where it sits until it's needed. And when it is needed, it's released into the small intestine and works directly in the small intestine. The pancreas, like the liver, also isn't directly involved in digestion, but what it does, which is vital, is it produces and secretes really essential digestive enzymes into the small intestine. The three key ones that you need to know about at GCSE: amylase, protease, lipase. More about those in a bit. The small intestine, the food has now passed from the stomach into the small intestine, and at this point, the food has already gone under a little bit of digestion using enzymes. Remember the amylase from the saliva in the mouth, and we also have the protease in the stomach. But it's still not fully digested. It needs further digestion. Once that food then enters the small intestine, more enzymes get secreted into that area from the pancreas and also from the lining of the small intestine too. Amylase, protease, lipase enzymes. So they help to carry on the chemical digestion of protein, of carbohydrates and fats in our food. And we need this to happen so that the food, by the time it gets to the end of the small intestine, which is a very long tube, it has been fully digested, i.e. large insoluble substances we've ingested, food we've ingested has become soluble, absorbable nutrients that can then enter our bloodstream. Once in the bloodstream, they can then be transported to our cells and used. Remember though, that there's always going to be parts of our food that we cannot digest. If we don't have the enzymes for that type of food, or that type of chemical or molecule like cellulose, fibre, we're not going to break it down. They will therefore have to be digested. That food enters the large intestine, and there's still water in there as well, which is useful to our bodies. The large intestine will absorb that water into the bloodstream, but obviously the undigested food is staying behind. It remains there because we can't absorb it. It's too big. That will then pass into the rectum for storage. It's a storage pouch, which is essentially now our poo, our feces. And then when we go to the toilet, we use the anus as the exit point for that undigested food to come out of us. We're going to focus now on how enzymes specifically get involved with the chemical part of digestion. Enzymes work using a model that we use a model to explain how they work, and it's called the lock and key model. We'll take a closer look then. First of all then, let's address, well, what actually is an enzyme? Enzymes are not living. They are in fact proteins that we call biological catalysts, meaning they work inside living things. That's the biology part. Catalysts, they speed up reactions. All catalysts generally speed up reactions, but these are specific to living things, hence that term. When they do carry out their reaction, they aren't actually used up themselves in the reaction, which is a great thing because it means they can be used again and again and again, as long as it's the same reaction type. The shape of an enzyme is extremely important because enzymes have a unique shape according to which substrate, which chemical they work with in the reaction. And we call this shape its tertiary structure or 3D structure. And that shape provides an area in the enzyme called the active site, which is very, very specific to the substrate that the enzyme reacts with. We therefore describe the enzyme as having specificity. It will only catalyse a very specific reaction. Let's have a closer look at that. The lock and the key theory then, or model. How does it work? How do we explain this? This is a simplified way of showing how enzymes work. The enzyme in this case is described as the lock. So if you think about a lock on your door, it has a very specific shape. And the area of the enzyme, which is most important here, is the active site. If we were to add this onto our diagram here, the active site is this area of the enzyme here. And that shape of the active site will only bind with a particular specific substrate that happens to complement its shape. The substrate is therefore described as being the key in this model. So as I've just said, the active site of the enzyme is a specific shape and only the right substrate will fit and we describe the substrate as being complementary. That's a really, really important term to use when you're describing how enzymes work using the lock and key. And also, once the binding has happened, where the substrate has bound to the active site of the enzyme, we call this the enzyme substrate complex. Another key phrase which is essential to use when you're answering exam questions related to the lock and key model. Once the enzyme substrate complex has formed, the reaction will happen and products will be made. And when the products are made, they literally will leave the active site because they're different, they no longer complement it, but the enzyme itself is unchanged and is ready to be used again. Now, a lot of questions in the GCSE require you to know how to measure how fast an enzyme-controlled reaction is happening, the rate of it, i.e. how quickly the enzyme converts its substrate into the products. And there's a variety of ways you can do this. Enzyme-controlled reactions can be affected by a multitude of environmental factors or factors involved in the reaction. I'm going to write a few of these down. Things that can affect the rate of an enzyme-controlled reaction typically would be the temperature of the reactants. Enzymes are very temperature sensitive. If it's way too hot for them, they will denature. If it's way too cold for them, they will lose kinetic energy and the reaction will slow down too much. Likewise, a pH change can have a massive impact on how fast an enzyme works, because enzymes are also very, very sensitive to pH and can denature in a pH that is not optimum for them. The enzyme concentration, as well as the substrate concentration, also have a big impact on the rate of the enzyme-controlled reaction. But how would we measure it? Let's say we wanted to investigate one of these factors and we were to choose one, like let's see how fast this enzyme will work if we change the temperature. How would you actually go about measuring the rate? You'd have to obviously collect something or look at something over time which shows us the reaction is happening. So it might be by collecting and measuring the product formed over time, or even the disappearance of the substrate, if there's some way you can see that over a period of time. And you'd have to measure how long that took, how long it took for the product to form, or how long it took for the substrate to disappear. So what would we do? We would have to use a mathematical application. The rate of reaction is going to be the change, for example, in the amount of product formed divided by the period of time it formed over. So for example, let's say you were carrying out an enzyme-controlled reaction where a gas was produced. So I'm going to give you an example of a reaction involving an enzyme. There is a substrate called hydrogen peroxide, H2O2, which is a chemical we produce naturally in our cells, but it's a toxic byproduct of our metabolism. We don't want it to be in our cells, but it is produced. But it's okay, because our cells have enzymes called catalase enzymes, which break down the hydrogen peroxide into harmless products, water and oxygen. Now, it's very, very difficult to collect the water because there's fluids if you've got all the reaction going on in a conical flask, for example, it's all fluid. How would you know how much that fluid was still hydrogen peroxide and how much of it was water? That would be impossible to measure. But you could measure the volume of gas produced by collecting the gas in a syringe, for example. So let's say you managed to collect 30 centimetre cubed of the oxygen gas in 10 seconds. If we go to the graph here, we can also use the graph to look at this. So in 10 seconds, if you're looking at time, that would be bang in the middle of the x-axis here. And then if you take that up to the curve and along, you can then get a basic idea. It's around 30. We're not being very precise here because I haven't got a ruler, but it'd be somewhere around 30 centimetre cubed of gas produced over that 10-second period. So you'd typically divide 30 by that 10, and that would give you 3 centimetre cubed per second. Now, there are actually other ways that you can calculate the rate of reaction using a graph, and this can be through using a tangent. So I'm just going to rub off my pen to try and show you how this is done if I can. It's not let me do it. The way you would do it is you would take a ruler. You'd have to make sure that the ruler was really straightly aligned with the steepest part of your curve. Now I'm doing this freehand, so I'm not going to get this really straight. But if you had your ruler on that line at the very steepest point and let it go off, then what you're actually doing is you're drawing a tangent. And what you'd then do is you'd go from that tangent where it goes off the actual line of the graph, you take a line down to your x-axis and you'd call this area of the tangent here y and this area of the tangent here x, and you do a very simple calculation. You'd do y over x. You'd have to know what each tiny square is worth, which you can work out because there's 1, 2, 3, 4, 5 mini squares in that 20. So 20 divided by 5 gives us 4. So we know that we've got 4 per mini square. So if we count that up, 4, 8, 12, 16, 20, 24, for example. So if it goes up to about 24 at the y part of the tangent, we then divide it by, now it's 8 here because it's 2 of those tiny squares. And that would also give us 3 centimetre cubed per second. So you can use a tangent on the graph if you want to get a much more precise reading. Generally, overall, the steeper, I should say, the gradient of the line, then the greater the rate of reaction is. Once the graph starts to level off, that's when the reaction is starting to slow down, and that's because the products have been made, the substrate's been used up. We're going to address now why enzymes are, or how enzymes are quite sensitive to the conditions around them. Now, I mentioned before that enzymes are sensitive to temperature changes and also to pH changes in their environment. We'll talk about temperature first, and then we'll move on to the pH and then look at 2 other factors too, the substrate and the enzyme concentration. What about temperature then? The graph on the slide is showing you that temperature is going up and up and up and up, and so is the rate of reaction at point 1 on the graph. And that's because as you heat the enzyme and its substrate up, they're both going to have more kinetic energy, which means they're going to move more frequently and they're going to collide more often, and enzyme substrate complexes are going to form more often. So the reaction is going to get faster and faster and faster as that temperature goes up. However, there is a point at which the enzyme reaches its optimum temperature, which means it's got to a point in the reaction where the temperature allows the reaction to happen at its fastest rate. And if you go beyond that temperature, that's when problems start to arise. Once the temperature gets too hot and goes beyond the optimum, then that can alter the shape of the enzyme because it causes the enzyme's bonds to break in its protein structure. And as the bonds break, the enzyme's shape changes, which changes its active site, and you can no longer get the substrate binding to it because the complementary shape of the substrate is no longer a thing. We don't have a complementary shaped substrate anymore to that enzyme because the active site is different in its shape. At that point, you can see on the graph, the enzyme controlled reaction, or the rate of reaction, drops dramatically. It's no longer happening because the enzyme is no longer functional. If we were to address the pH as something that can affect enzymes and their reaction, it's a similar idea. Enzymes are very sensitive to pH in their environment, but it's not a set in stone pH. Not every enzyme has an optimum pH of 2 or 7 or 14. Different enzymes have different optimum pHs depending upon where they work. For example, protease enzymes in our stomach will work at their optimum at a pH 2, and you can see this on this graph here that pepsin, the green line on the graph, is showing an optimum, a peak in the reaction, the activity at around 2. Whereas amylase in the small intestine is going to work at its best at a more neutral pH, almost slightly alkaline, 7.5, and we can see that that's been highlighted here on the graph already for you. If the pH does get too high or too low for that enzyme, then that enzyme will also go into a denatured state because it causes bonds to break in the enzyme's shape, enzyme structure. Pepsin, therefore, would denature if you put it in a pH of 4. Amylase would denature if you put it in a pH of 3 or a pH of 11. You can see that on the graph. So just to reiterate, denaturing is when the active site changes shape, the enzyme substrate complexes can no longer form, and the reaction will come to a halt. Now, enzymes come in all sorts of different forms, and some are more tolerant than others. Some will have on the curve for their pH sensitivity a much wider tolerance. So if we look at amylase, amylase has a much wider tolerance for pH change than, say, the blue line, which is catalase in this example. So all enzymes that exist are quite different to each other as to what conditions they will work best in, and also what they can tolerate before denaturing takes place. And then we can just see, as the slide says, the graph is just showing the optimum pHs for our 3 different enzymes there. The other 2 factors that can affect the rate of an enzyme-controlled reaction is, well, how much substrate there is to collide with the enzyme. Now, eventually, as the substrate gets converted to product, if there's no more substrate left, then of course you're going to run out of substrate for the enzyme to react with. Or it might be that you're limited in terms of how much enzyme is present. So you might have loads and loads of substrate, but there's not enough enzyme available, or the active sites are occupied for the substrate to all be broken down at that point in time. And at that point, you will get a constant rate of reaction. So your graph will show the enzyme rate of reaction going up, but it plateaus, that means flattens, when there comes a point where the reaction can't get any faster, and that would be when you've got active sites fully occupied, even if you have lots and lots of substrate still waiting around to react with the enzyme. Enzyme concentration as well will have an impact. So if you add more enzyme, there's more active sites available for a faster reaction, but the substrate might run out. If you don't increase the amount of substrate and the substrate becomes limited, then adding more substrate is not going to have any further impact on the enzyme controlled reaction, even though there's enzyme available. So let's summarise our digestive enzymes. You do need to know these for your GCSE. You need to know our 3 big examples: amylase, protease, lipase. You need to know the substrate that they react with. Amylase reacts with starch, which is a type of carbohydrate. Protease reacts with protein, and lipase reacts with lipids. So amylase breaks down starch or catalyzes that reaction. Protease, protein. Lipase, lipids or fats. Make sure as well, guys, that you know the products that come from those 3 different reaction types. From starch being broken down, we get simple sugars like glucose. From protein, we get amino acids. And from fats, we get glycerol and fatty acids. The other things, there's quite a lot to learn here, guys, so it's always good to have a document somewhere that you could look at regularly just to keep revisiting it. You need to know where the different enzymes come from in the body, and you also need to know the site of action, so where they act, because they might be produced in a certain place, but they don't act there. Let's go back over them again. Amylase comes from the salivary glands in your mouth. The pancreas secretes enzymes into the small intestine, and the small intestine lining itself secretes these enzymes. Where this enzyme acts is in food as it enters the mouth and as food enters the small intestine. Protease comes from our stomach lining. again from the pancreas and from the lining of the small intestine, and where it acts is on food present in the stomach or the small intestine. Lipase, I should say, comes from our pancreas, it comes from our small intestine lining, but it only acts on fats as they enter the small intestine. Facts, guys, you do need to learn for your GCSE. What do we do then with these products of digestion? Why do we need them? Why do we need to absorb sugar, glucose into our bloodstream? It's because they need to be delivered to our cells, because glucose is our respiratory substrate. We use it in respiration. We use it to release energy to allow us to carry out all our life processes. Glucose can also be built back up again into complex carbohydrates. For us animals, we tend to build our glucose up into glycogen, which is a storage carb in our liver and muscle cells that we can then go back to and use later on should we need glucose from it. Amino acids. We need amino acids to build proteins. We call this protein synthesis. Lots of parts of our body are made of protein. Our muscles are a good example of that, as are our enzymes. Fatty acids and glycerol, we build these back up inside our cells into lipids again. Fat is an essential part of our diet. Lots of things rely on fat. Our cell membranes are made of fat. We also have oils in our skin, which are fat-based. We also need oils to kind of lubricate areas of our body too, like synovial fluids, things like that. Synovial fluid is kind of fluid found in our joint area. And a lot of our hormones are fat-based. Therefore, these products of digestion are essential to life. Let's just go back to the liver then and bile. We talked about this right at the beginning of the video, but we didn't really go into a huge amount of detail on what bile is. Bile is a yellowy green fluid which is made by liver cells, and when the liver makes the bile, it then stores it in a little pouch which is found located just under the liver called the gallbladder. When food is in your small intestine, the gallbladder releases that bile into the small intestine where it works on the fat. Now, it actually has another function as well. That is actually a really important function. It helps to neutralize the acidity of the food coming out of the stomach into the small intestine. Remember that in the stomach you have a really, really low pH of around 1 to 2 because hydrochloric acid is produced in the stomach for the protease enzyme to work at its optimum. But that acidity is not going to be good for the enzymes of the small intestine. They prefer it a little bit more neutral, even slightly alkaline. What this bile does is it helps to neutralize that acid in the food as it enters the small intestine and provides an optimum pH for the enzymes of the small intestine so they work effectively and efficiently around pH 7 to 8. The second really important function of bile is a process called emulsification. We can see this in the diagram over here. Emulsifying fat is not a chemical process, it's a physical process where large fat droplets are literally kind of separated out into smaller fat droplets. I often use the scenario of if you've just done some frying in a frying pan and you've got all these fat globules that you want to wash away because you want to wash your frying pan, you put in some washing up liquid and that disperses the big fat droplets into smaller fat droplets, making the pan easier to clean. That's like an emulsification. And that's what bile is doing. It's breaking down, or breaking apart, because it's not a chemical reaction, fats into smaller droplets. Now that is a really, really important thing because that overall increases the surface area of the fat, enabling the lipase enzyme to work more quickly. If there's more surface on these smaller fat droplets available for the enzyme lipase, then fat digestion is going to be faster. This therefore, just to summarise that, it increases the rate at which the lipase enzyme digests lipids into fatty acids and glycerol. Moving swiftly on, Now, part of this module in GCSE has a couple of required practicals which you can be asked about in your exam. One of them is the qualitative reagent food tests, where you literally test food samples to see if they have in them types of carbohydrates, starches and sugars, also looking for proteins and also looking for fat in food samples. We're just going to quickly run through what these tests involve. You've got to know the reagent, what you're looking for, and what result you would see if that food chemical was present. Before we start though, just to mention that we call these qualitative tests because what they're doing is they're just allowing us to see the presence of a molecule. We're not actually testing how much is there. That would then be a quantitative test. We're not doing that. We're just looking at the quality of the test. Is that biological molecule present or is it not? The first test is the Benedict's test. What are we testing for here? We're looking for sugar in food. But the bit of detail method that's vital to mention if you're asked about Benedict's test in an exam is that it requires heat, quite a hot water bath. We're talking, 80 degrees C plus, that the reaction has to happen in. So you'd place your food substance, your biological molecule, into a test tube. You'd add your Benedict's reagent, which is a blue reagent, and you'd plonk it in a hot water bath for about 5 minutes and wait. Now, the colour change that takes place, if sugar is present, depends upon the concentration of the sugar present. If we look at this chart here, it's showing you changes in intensity of colour that the Benedict's turns with the sugar depending upon the concentration of the sugar. Now, if there's no sugar at all, you're not going to get any reaction. So you're going to still see a blue tube because you're just going to see Benedict's still as Benedict's. If sugar is present, but only a trace amount of sugar is present, you are going to get a slight colour change to a green or a yellow. If there's a moderate amount of sugar present, then the colour change is a little bit more orange. And if there's a lot of sugar there, you're going to get a really nice brick red colour in your results, showing that you've got a lot of sugar present in that particular test tube. The starch test. Starch is a carbohydrate as well as sugar, but starch is a different type of carbohydrate. It's a polymer, not a monomer. To test for starch, you would use a different reagent. This time we would use iodine. Iodine is orange. So a negative test, if you tested a chemical for starch with iodine, when starch isn't present, you'd still get an orange colour. But if starch is there, the iodine will react immediately with it and turn it blue-black. And that is a positive indication that starch is present in that test tube. Protein uses a reagent called the biuret reagent, which is blue. It's like a nice pale blue. And when you mix it with a sample of food that contains protein, it will go to purple or lilac. If there's no protein there, it just stays blue. That actually should say protein there, and so should that. So we've got our negative test where protein is absent and our positive test where protein is present. And the last food test is our lipid test. Now, lipids can be tested using ethanol, which is a type of alcohol, and water, and we call this the emulsion test. You take your sample of food to be tested, you'd add ethanol to your sample and shake it, you'd then add some water into it and look for a cloudy white emulsion. So you can see the steps here. Here's your sample to be tested, you've added your ethanol, you've shook it up, you've then added your water and it should go white. So this is your positive result at the end of the emulsion test. Our second required practical is a little bit more technical than just looking at the qualitative test one. This time we're going to investigate the rate of an enzyme-controlled reaction if we change a particular factor that we know can affect enzymes. And in this example, it's pH. We know that enzymes are pH sensitive, so can we put enzymes and their substrate in different pH environments and see if it has an effect on the rate of the reaction? Now, to do this, The procedure is quite technical, so you have to really think about what you're doing. First of all, we're going to set up our water bath at a temperature of typically 37 degrees C. Why are we doing this? We don't want the temperature to also be a variable that impacts the rate of the enzyme controlled reaction. Therefore, this is one of our control variables. We need to keep this the same throughout the experiment, so we know that any effect on the rate of the enzyme reaction is to do with the pH only. The water bath is set up at a temperature that is typical of most enzymes' optimum temperature and also typical of human body temperature. Next thing you do is you choose obviously which enzyme and which substrate you're going to use in your experiment. We're going to use the amylase enzyme and starch, because starch is the substrate to amylase, and the indicator of presence of starch is iodine. So that's going to be a reagent that we use in the experiment to show if things are happening or not. We're going to put the iodine into a spotting tile. So you can see in the picture here, we have a spotting tile which has in it, in each little well, it has a little bit of that iodine. Once you've done that, we are then going to take our enzyme, and our enzyme, remember, is amylase, and we're going to put the amylase enzyme with a buffer of known pH in a test tube and the substrate starch into another test tube. Let me explain a little bit more. Amylase will work at an optimum pH, so we're going to set up a variety of different pHs to find out what that is. Buffers are chemicals of set pHs. We have different buffers. Some buffers are pH 7, some are pH 4, some are pH 8, and so forth and so on. So what a buffer solution is it's a solution of a known pH. We're going to mix the enzyme with a buffer solution of a chosen pH so that the enzyme is thoroughly mixed with that pH and set at that pH in its environment. In the meantime, the substrate that the amylase works with in the reaction, starch, is put into another test tube. We don't want them to mix yet, or the reaction is going to happen. Before we mix them together, what's really important is that we equilibrate both of these tubes to the same 37 degrees C temperature. So the two separate tubes are going to be put into the water bath of 37 degrees C and left for a little while. You can check the temperature with a thermometer in order to determine at what point they both reach the same temperature, and then you can react them together. Obviously, if you had one at room temperature and one at 37, then when you pour them together, that's going to change the temperature. So we need to make sure they're both at 37 to make sure that that is a standardized variable, and that's not going to have an impact on the rate of the reaction. So here we have our two tubes at the side. We have our tube with our amylase solution in and our buffer of chosen pH, and we have our substrate starch. Once they have reached the same temperature as each other, and we have checked that, we're going to pour the starch into the amylase. But as soon as you do that, you're going to have a reaction probably happening if the pH is a pH the enzyme can work at. So when you mix them together, you must immediately start a stop clock. And the best way to do this, the most precise way to do this, is to measure in seconds, a small unit of time, and look for your results every 10 seconds. But how are we going to look for those results? We're going to take a sample of our mixed enzyme and starch with a pipette and drop it into a well of the iodine solution. Now, the iodine solution will go blue-black if the starch is still intact and the enzyme hasn't yet acted upon it. If the iodine solution goes orange-brown, then the reaction has happened. So that's the observable colour change we're going to use to determine when the reaction has taken place. So the reaction will have taken place if we see orange-brown, if the starch is no longer present to react with the iodine. Let's have a look at how this would work. So here are our wells of iodine over here. When we put the first drop of our enzyme substrate mixture into the first well, it stayed blue-black at 10 seconds, telling us that the reaction hadn't yet happened. Then at 20 seconds, again, no reaction. At 30 seconds, no reaction. At 40 seconds, no reaction. But then 50 seconds later, we start to see a change. And then by 60 seconds, there is no starch left in that test tube because the amylase must have broken it down. So what if this pH buffer that we used had been pH 7, then we know it took 60 seconds for the reaction to happen at that particular pH. And this is basically the way we're going to carry out the experiment over and over and over again, but at different buffer pHs. So just to reiterate, you're going to record the time it takes in seconds, every 10 seconds, for the iodine solution to stop turning blue-black, i.e. when the starch has been broken down into sugar. You then repeat the experiment starting from scratch, but using a different buffer each time. So you could choose a nice range like pH 5, pH 9, pH 14, as well as the pH 7 that you've just investigated. Let's have a look at what results might look like. So at pH 5, it took 80 seconds for the starch to no longer be present. So 10, 20, 30, 40, 50, 60, 70, 80. This was the point where we have seen a reaction taking place. At pH 9, it took 90 seconds. 10, 20, 30, 40, 50, 60, 70, 80, 90. And then at pH 14, nothing happened. The starch stayed as starch. There was no indication of any reaction because the iodine kept turning blue-black, telling us starch was present throughout. So pH 14 must have been a pH that denatured the enzyme when it was mixed with the enzyme at the beginning of the experiment. So that is our typical method to how you would carry out this practical. But there are other things that you do have to look at and you could be asked about, i.e. what were the variables involved? What is the independent variable in this investigation? The independent variable in any experiment is the one that you change. It's the thing that you're investigating. Is this thing having an effect on your results? In our experiment we've just discussed, it was the pH. That was our independent variable. We changed that. Dependent variables are our results. So what are we recording as a result? In this case, it was the time taken in seconds for the starch to be digested, for the iodine to stop going blue-black. The control variable, or control variables, are all the variables that could affect the enzyme-controlled reaction, but we don't want them to. We want to keep them standardized so we know it's only our independent variable that's having an effect. The typical examples would be keep the temperature the same, which we did with the water bath, and all your volumes need to be the same each time. You can measure that with a measuring cylinder or a pipette or even a syringe, and also use the same enzyme and substrate concentrations throughout. Now, you can carry out this experiment with different independent variables. So instead of changing the pH, you could alternatively have used the same pH. You could pick the optimum pH for that enzyme, like pH 7. Make sure that the buffer throughout is used as pH 7. And you actually put your tubes of amylase and starch at different temperatures in different water baths. But you would still carry out the practical in the same way, putting drops of the mixture into the iodine to see at what point the starch disappears. So you can twist the experiment in different ways to investigate different factors that affect the enzyme's rate of reaction. A couple of other little important points to note that could come up in questions. Sometimes the exam will ask you to say, how could the precision of the experiment be improved? Or how could you make your experiment more accurate? We often get quite confused with our key scientific words. What does this mean then? If we're looking for more precision, then this is about always repeating your experiment as many times as you have time for. It must be at least 3 times. If you only do an experiment twice and you get 2 completely different results, you don't know which one is right and which one is wrong. So you'd have to keep doing it until you see a pattern. You get the same results each time. Obviously, you might get the odd set of results that look weird. We'd call them the anomalous results, and they have to be removed. You wouldn't include them in your results before you calculate any kind of means. If we're thinking about accuracy, accuracy is about basically using a measurement of smallest value as possible. In this case, we're investigating what the optimum pH is. There's no point just looking at pH 2 and pH 6. It's not very accurate because we don't really know if there's any in between that could have been better than 2 or 6. So we do as many pHs at as small intervals as possible to get a nice accurate idea of which pH is the optimum for the enzyme controlled reaction to happen at its fastest. Next part of our organisation we're going to focus on is our respiratory system. We're going to focus on the lungs and how air enters our bodies in this part of the video. We can see here on the slide we've got a breakdown of all the different parts of our respiratory system that are involved, and we're going to go through each one and make sure we've got a good understanding behind it. Let's just start with generally what the lungs are. The lungs are parts of the respiratory system which are highly adapted for gas exchange. Gas exchange is when oxygen is exchanged with carbon dioxide at the alveolus, at the air sac. Oxygen has to be able to enter our bloodstream and hence delivered to our cells, and carbon dioxide has to be able to leave our bloodstream and then be exhaled. We don't want carbon dioxide to stay in our body in high levels because it can be quite toxic. We're going to focus on how our body does all of this from start to finish. Starting with the pathway that air travels through once it enters your lungs. Now, there are a variety of tubes that air has to pass through to reach the point of gas exchange, which is at the alveolus. And it all starts at the trachea. Obviously, air has to be breathed in through the nose or through the mouth. And when air is breathed in through the nose, it's actually better to breathe in through the nose because you've got hairs there that can filter the air. You've also got lots of blood vessels lining the nose, which warms the air as it passes through. The air will then travel down into the windpipe, otherwise known as the trachea. You can call it a windpipe as well. And that trachea, which is held open with cartilage, we can see on the diagram these little rings of cartilage which hold it open, and the air will travel down them until it reaches a point where the trachea subdivides into 2 bronchi, which is around here. And those 2 bronchi, the subdivisions of the trachea, take the air off towards each lung, but the bronchioles subdivide further and further and further into tiny bronchioles, which then branch into each lung, terminating in these tiny air sacs, which look like little kind of areas of grapes or bunches of grapes, and that's where the gas exchange takes place. It's important that you know the names of these different structures and you can identify them on a diagram too. The lungs themselves are surrounded by the rib cage, which are your bones, and there are muscles between the ribs, and you also have a muscle at the base of the chest cavity called the diaphragm, and this is a very, very important muscle needed when we ventilate. That's when we breathe in and out. The ribs are obviously a protective cage, but they also are involved in ventilation, both very important for that process. We're going to focus then on the mechanism of how we actually breathe in and out, and it's all to do with the rib cage and the diaphragm. The lungs themselves don't just inflate and deflate independently. There has to be something that pulls air into the lungs and pushes air back out of the lungs. Now, the mechanism of breathing is called ventilation as well, and as I've just said, it's about moving air in and out of the lungs so that the air reaches the alveoli, and air that's old air, i.e. air with lots of CO2 in, is removed from the alveoli. This process involves muscle contractions, which means that it's active. It's a process that requires energy because muscles need to contract in order for you to ventilate. How do we inhale or breathe in? Inhaling or breathing in involves the muscles between the ribs. Now these are called the that means between costals, that's a technical name for rib. The intercostal muscles contract. We can see on the diagram that the ribs here, you can't actually see the muscles of the ribs on this diagram, but in between there would be muscle, as I'm drawing in now. When those muscles contract, the rib cage is pulled up and outward. The diaphragm contracts at the same time, and as the diaphragm contracts, it flattens, it moves downwardly. Now, the rib cage moving out and the diaphragm flattening effectively increases the volume inside your chest cavity, and that drops the pressure inside the lungs. So the pressure outside your body is now greater than inside, so we get a pressure gradient where that pressure gradient causes air to move down the pressure gradient from high pressure outside your body to lower pressure inside your lungs, filling your lungs with fresh oxygenated air. So oxygen will reach the alveoli through that breathing in or inhalation. The opposite to that is exhalation or breathing out, and it's really just inhalation backwards, if you like. This time the diaphragm relaxes. When the diaphragm relaxes, it pings up into a dome shape. And when the intercostal muscles relax, the rib cage drops, so it moves down and in. Now, collectively, those two changes drop the volume inside your thorax, which makes the pressure go up inside. So there's higher pressure now inside your lungs than outside your body, which literally forces the air back out again, down that pressure gradient from high pressure to lower pressure. So that is how we breathe in and breathe out. And it is vital, guys, that you can explain that mechanism using the diaphragm and the intercostal muscles and what they do in your description. We're going to zoom in on an alveolus now. The alveolus are the points for gas exchange. They are the tiny air sacs that we find in bunches at the end of the bronchioles. There are millions of these in each lung. We have a huge quantity of them, which makes the surface area of our lungs absolutely massive. In fact, the surface area of our lungs can cover the entire surface of a tennis court. It is that big, which means our gas exchange is super efficient, super fast. Now, the alveolus are the important part of gas exchange because this is where the diffusion of the gases take place of oxygen and carbon dioxide. Oxygen moving into the blood from the air you've just inhaled, and carbon dioxide moving out of the blood so that you can exhale it. How are the alveoli super adapted for this process to be efficient? Well, first of all, we've just mentioned this, we have tons and tons of alveoli. So collectively, they cover a very large surface area, which maximizes the rate of diffusion. Now, each alveolus, if you look at the diagram here, the lining of it is one cell thick. So there's only one cell that lines each alveolus. Now, the capillary, which is this tube around the alveolus, that's the blood vessel. I'm going to label that in actually, that's a capillary blood vessel. The capillary is also only one cell thick. So in fact, the gases moving in and out of the blood in the alveolus, so CO2 is moving back out and oxygen is moving into the blood. They only have to pass through two cells to do that, and that's very small distance to travel. Therefore, this one cell thick alveolar lining and this one cell thick capillary lining provides what we call a short diffusion distance. There's not much distance over which the gases have to travel to get to their destination. Now, on this diagram, we're only seeing one capillary, but in reality, each alveolus has tons of capillaries associated with it. So all these capillaries mean that blood is constantly flowing into the lungs and out of the lungs. It's moving all the time. So as soon as oxygen gets into the blood, it's moved away and is replaced by deoxygenated blood. So that helps to maintain a steep concentration gradient for oxygen and carbon dioxide to ensure they keep diffusing from a high to a low concentration. Okay, now we've obviously addressed how the lungs are working in order to bring oxygen into the blood and get carbon dioxide out of the blood. But what the respiratory system works in conjunction with really effectively is our circulatory system. What is our circulation or circulatory system? The circulatory system is the system that then transports the oxygen away from the lungs to the body, to the cells of the body, or it brings carbon dioxide rich blood back to the lungs for exhaling and getting rid of the carbon dioxide. Also, don't forget that the blood transports a multitude of other things around our body. It gets the nutrients from the small intestine that we've absorbed and takes them to the cells. It also transports hormones from glands to target organs to have their effect. There are main things that make up our circulatory system. We have our heart, we have our blood vessels, and we have the blood, and they work as a collective, making this system really, really effective for its job, or in its job. Before we go into the details of the heart, the blood vessels, and the blood, the other important thing to address is that in us mammals, we have what we call the double circulatory system, which means that our blood passes through our heart twice on each complete journey around the body. But there's two circuits going on working at the same time. We say that they are working simultaneously. And the two systems or circulatory systems working simultaneously are called the pulmonary circulation and the systemic circulation. Pulmonary generally in biology means lungs, and systemic means your system, your whole body. We're going to focus a bit more on our next slide on what we mean by that. Should be an L. Right. Pulmonary circulation. This is when your deoxygenated blood is pumped from your right side of your heart, to your lungs. So deoxygenated blood which has no longer got oxygen in it, moves from the right side of your heart, but it is pumped from the heart to the lungs to become oxygenated. So once that blood gets to the lungs, gas exchange takes place. Oxygen enters the blood and any carbon dioxide in the blood enters the lungs. This is all a diffusion process, by the way, of the gases moving between the blood and the alveoli. Once the blood has become oxygenated at the lungs, the unfortunate thing about the blood, when it leaves the lungs, it's lost a lot of pressure behind it, so it's not going to move very fast. So what the pulmonary circulation involves is taking that blood from the lungs, now oxygenated, back to the heart to get a pressure behind it so it can be pumped right around the body. But it returns to the other side of the heart, the left side of the heart, from the lungs. In terms of the systemic circulation, once this oxygenated blood has returned to the heart from the lungs, the heart then, from its left side, pumps that oxygenated blood all the way around the rest of the body. So the oxygen is delivered to all the cells for respiration, but in doing that, the blood becomes deoxygenated again and then goes back to the heart again on the right side to be pumped to the lungs again and enter the pulmonary circulation. So it's like two circuits going on at once, from heart to lungs to heart, heart to body to heart, heart to lungs to heart, heart to body to heart. And that double circulation ensures we have enough oxygen travelling around our body all the time, and we are also having enough pressure behind the blood to move it to where it needs to be at the right speed. Now, I've kind of just said this, so why is double circulation so important? We obviously want to ensure we maintain a high pressure in the systemic circulation, so oxygen-rich blood reaches all of our body tissues very quickly, and it also ensures that oxygenated and deoxygenated blood do not mix, and that means we get fully oxygenated blood going to the right places and only deoxygenated blood going to the lungs to become oxygenated. It makes it very efficient. We'll look at the heart now and how it is structured. You do need to know how to label the heart and the different vessels that come from the different or come into the different parts of the heart. Remember that this heart is part of a double circulatory system. Some animals don't have the same types of hearts as us. For example, fish only have 2 chambers in their heart, us mammals have 4. We have a double circulation because of that, whereas fish have a single circulation. So don't assume that every animal has double circulation. Ones that are relatively big, animals like us, do need the double circulation to make it more of an efficient circulatory system. Let's focus on the chambers first of all. The thing to remember, guys, when you look at a heart picture, you're looking at a mirror image of it. So as you're looking at this picture, you're looking at the right side as though it's your left, and you're looking at the left side as though it's your right. It's always the mirror image. You've got to remember that. Now, the right atrium, the right side here, is our top right chamber. This is the chamber that's receiving the deoxygenated blood that's just come from the body, so it's full of carbon dioxide. And it enters the right atrium through a really big vein called the vena cava. This is a vein. And there's actually 2 bits to the vena cava you can see here, because the top vena cava, or superior vena cava, brings blood from the kind of top regions of the body, like the head and the neck area and the arms, whereas the vena cava branch here, which is inferior, brings blood back from the lower parts of the body into the heart, into that right atrium. The right ventricle is directly below the right atrium, and its job, its role, is to pump the blood to the lungs, and it pumps it out through a big artery called the pulmonary. Pulmonary in biology means lung, so that's going off to the lungs to get oxygen, that blood there, so it's coming out here. And we have 2 branches coming off of that because there's 2 lungs that need to receive blood via the pulmonary artery. Just a bit of a fact here, this is the only artery in our body that has deoxygenated blood in it. All other arteries have oxygenated blood in them. The left atrium, remember, the blood has gone to the lungs and picked up oxygen. It's going to come back to the heart because it needs pressure behind it to keep flowing. So it comes back into the left side, into the left atrium, through the pulmonary vein. Now, this is the only vein that has oxygenated blood in it. All other veins in the body are deoxygenated. I'll talk about arteries and veins later and why they're different. So the left atrium is receiving oxygenated blood from the lungs via the pulmonary vein. The blood is then pumped down into the left ventricle, and the left ventricle is responsible for then pumping that blood up and out into the big major artery of our body called the aorta, and the aorta then takes that blood off to all the different parts of the body, i.e. the systemic circulation. The other question that comes up regularly is why the left ventricle has a thicker muscular wall than the right, and it's because of how much pressure is needed to push that blood to where it needs to go. It's logical. The right side, the right ventricle, is pushing blood when it pumps, when it contracts, to the lungs, which is right next to the heart, so there isn't much distance that blood has to travel to reach its destination. On the other hand, the left ventricle has to be able to pump the blood all the way around the systemic circulation, all the way around the body, and that takes a lot of pressure. So the left ventricular wall has a thicker muscle component, it's much thicker in its muscle, in order to contract more forcefully and pump the blood at a higher pressure to get all the way around the systemic circulation. The pulmonary circulation does not require the same pressure. We'll just revisit those blood vessels then, and a few others. Blood vessels that are associated with the heart. We've mentioned the first 4 on my list here, but it is important, guys, that you can label the different blood vessels on a heart diagram and you know which direction the blood is travelling through them from or to. Vena cava, deoxygenated blood from the body to the right atrium. Pulmonary artery, deoxygenated blood to the lungs. Pulmonary vein, oxygenated blood from the lungs to the left atrium. The aorta is blood, oxygenated blood from the left ventricle to the body. We've addressed them a few times already, but what we haven't talked about is another artery type which is associated with the heart. This doesn't bring blood into the heart or out of the heart. What the coronary arteries are branches off the aorta that then wrap around the heart muscle itself and bring the heart a blood supply, because the heart is a muscle, it's very active, it needs oxygen, it needs glucose, it needs a really good blood supply, and that's what the coronary arteries are there to provide. Another part of the heart you could be asked about or be expected to label are the valves in the heart. Now, valves are little structures inside blood vessels or inside the heart that can open and close to maintain one directional flow of blood through the heart or through a blood vessel. Now, you can see here there's a valve here, and there's also valves at the bases of the two main arteries leaving the heart. When blood pushes on the valves in the right direction, it opens them so the blood can flow through. But if the blood tries to backflow, then those valves close, they snap shut in order to prevent blood going in the wrong direction. So if you're asked what the valves are for, this is your exam answer. They maintain a unidirectional flow of blood by closing. They close in order to ensure that that happens. Let's move on to our heartbeat and how our heartbeat is regulated. Now, we have in our heart a natural group of cells which are located in the wall of our right atrium. Here's our right atrium, and located in the wall of our right atrium is a little bundle of specialized cells which are known as the pacemaker cells, or the SAN, the sinoatrial node. And these cells are responsible for generating electrical signals that then pass across the heart and control the heart's rate of contraction. Sometimes people's pacemakers, natural pacemakers, go wrong. There's a fault in them, and they end up with an irregular heartbeat, too fast, too slow, or one that's just all over the place. And if that happens, then people have to have an artificial pacemaker put into their heart or put onto their chest in order to put the rhythm back to what it should be. Otherwise, it can actually be detrimental to survival. What is cardiac output? Cardiac output is the volume of blood that your heart pumps out per minute. Now, people have slightly different cardiac outputs depending upon your level of fitness. If you're athletic, your cardiac output tends to be greater because, just like with any muscle in the body, exercise increases your muscle strength. Your heart will be stronger if you are athletic and trained, and therefore your cardiac output will be greater. You can work out a cardiac output from somebody. This would be in the interest of a doctor, for example, who was monitoring somebody's heart health. What they would do, they'd use special devices in order to measure the stroke volume and the heart rate of the individual in question. But what does that actually mean? The stroke volume is the amount of blood, the volume of blood that is pumped out of one ventricle in one beat. They could use a scan to do this, to determine how much blood is actually leaving the ventricle each time the heart beats. They would also record the number of beats for the heart for that person per minute, beats per minute, and then use the two figures in a simple calculation in order to get somebody's cardiac output. So here's a typical example. If a person's stroke volume was measured by a doctor to be 75 centimetre cubed and their heart rate was 80 BPM, beats per minute, times those two together, and that person's cardiac output would come to 6,000. Don't forget units though. You've also got to include both units in your answer. Often they will ask you to make sure you include units If they don't, try and remember, because you can get an extra marking point for correct units placed after a calculation. We're going to look now at our different blood vessel types in a bit more detail. There are three main blood vessel types that exist in our bodies, and they all have their own very specific structure, i.e. they are adapted for their own very specific function. Now here's quite a detailed diagram of our different types of blood vessels. The main three: arteries, veins and capillaries. Now the arteries, the veins and the capillaries all have quite different functions in terms of where the blood is flowing to or from, or what's happening to the blood, or what type of blood is in it. Generally speaking, we're going to keep this quite short and simple. Arteries carry blood away from the heart. Veins carry blood towards the heart, and capillaries are between the arteries and the veins, and that is where exchange takes place between the blood and the tissues, the cells. The pressure in these vessels is all to do with how far away from the heart they are. Typically, arteries come away from the heart, so the blood pressure is high. Veins take blood away from the organs, so the pressure has dropped dramatically at that point, so it's lower. The capillaries are really, really small, and the blood kind of disperses through them and slows down as it moves. The pressure drops dramatically in the capillaries, and we need that slow movement so that exchange can happen. The wall thickness is according to how much pressure the blood is under, because we don't want our blood vessels to burst, for example. Arteries have thick muscle and elastic walls to maintain high pressure and to withstand high pressure. Veins have thinner walls because they don't have as much pressure inside of them, because the blood is coming away from the organs, so the pressure has dropped dramatically. The capillaries are only 1 cell thick because they are the point of exchange between the blood and the tissues. We need a short diffusion distance for oxygen, carbon dioxide, nutrients to move in between the blood and the cells there. The lumen size. The lumen is the hole in the middle through which the blood is travelling. The lumen is going to be much narrower in an artery because the elastic tissue bounces back. If the artery kind of expands a bit, stretches I should say, because blood is flowing through it quickly, it'll bounce back to its original size, and that helps to maintain pressure. Veins generally have a wider lumen, and capillaries are teeny tiny little blood vessels. They have a tiny lumen for one blood cell movement at a time to ensure that every red blood cell can exchange oxygen and carbon dioxide. Oxygen into the tissues, carbon dioxide coming back into the blood. Some of the carbon dioxide will attach to red blood cells, but most will flow into most carbon dioxide will end up in the blood plasma. Valves are there to prevent backflow. You're generally only going to find valves in veins, because veins are under low pressure and the blood has a tendency to go back on itself because of that, but these shut to prevent that from happening. So valves in veins, yes. Oxygen content: apart from the pulmonary artery, then arteries carry oxygenated blood to the organs, because remember that blood is coming from the left side of the heart, which has received blood from the lungs. The veins carry blood away from the organs, so the oxygen has been used up by the organs and therefore it will be deoxygenated. And the capillaries will have a mix of blood because they're in between. That's where all the exchange is happening. So CO2 will be moving into the blood, oxygen will be moving out of the blood at the capillaries. Now, if we're going to just summarise this, if you were asked in an exam to differentiate between your 3 types of blood vessels, you can keep it quite short and sweet. And these are the 3 main things you would say. Arteries: thick muscular walls to withstand high pressure, elastic fibres to stretch and recoil with the heartbeat. Veins have a wide lumen to ensure lots of blood can flow easily through them because the pressure isn't there that there is in the arteries. And the valves prevent the blood backflowing, because it can do, because it's under low pressure. And the capillaries have these very thin walls, they're just 1 cell thick, for rapid diffusion of oxygen, glucose and carbon dioxide. At the bottom here we have a picture of an artery in cross section. In fact, this is an artery again, it should be a vein, but just so you know that's an artery as well, it's just a mistake on the picture. And this is a capillary, just showing you the difference between our 2 blood vessels. If there was a vein here, then you would see a lot less elastic tissue in the walls and the lumen would be bigger. How would we calculate blood flow? Calculating blood flow is getting to know the volume of blood that's passing through a blood vessel in a unit of time. There's two big influences to this: how much pressure the blood is under and how much resistance the blood is experiencing as it flows. As blood flows through the blood vessel, it's encountering blood vessel wall, and that can slow the blood flow down. We call that resistance. There is a formula that you can use. They will often give you this in an exam, and then you have to put figures in the right place to work it out. Blood flow, what distance in what volume has moved in centimetre cubed in a time. So you'd get the volume of blood and you divide it by the time taken. So if they gave you an exam question which asked you to calculate blood flow, where a student had investigated it and found that 90 centimetre cubed of blood had flowed through a blood vessel in 3 minutes, you would just plug those numbers into the formula. 90 divided by 3 is 30, but don't forget your units again. You often get an extra mark for including those units. So it's centimetre cubed per minute. Blood then, blood as a tissue. Remember what a tissue is from the beginning of the video. A tissue is a collection of similar cells working together to perform a function, and the blood is typical of this. Blood is made up of different components, and each of those components has its own unique job. What we have on the slide just above me here is a test tube showing you that if you were to separate blood out into its components, obviously when we take blood from somebody's blood vessel, it just looks like a red liquid, a thick red liquid, because it's all mixed up. But in labs that test blood, they can separate it out into its different components. And blood is made up of 3 major parts: plasma, white blood cells and platelets, and red blood cells. And you can see on my diagram above my head there the general percentage within blood that each of those are at. But what are the functions of these different components? Blood plasma is the liquid of the blood. In fact, if you separate it from the cells, it's yellow. And in this liquid are all the dissolved substances that we need transporting around the body. Things like carbon dioxide to get it back to the lungs to breathe it out, urea in order to get it to the kidneys to be excreted. We also have hormones moving from glands to target organs, and nutrients from our small intestine to our cells. The red blood cells carry the oxygen, and they use a very special protein called hemoglobin to do that. Hemoglobin is part of the red blood cell that oxygen attaches to, and therefore the oxygen is carried really efficiently to the cells for their respiration. Our white blood cells are our defense cells. They're there to help us fight disease, pathogens like bacteria, viruses, and there are different types of white blood cells. Some carry out phagocytosis and some produce antibodies. And then the platelets. Platelets are actually fractions of blood cells, they're not full blood cells as such, but what they do is they get involved with our blood clotting mechanism and therefore prevent bleeding at the site of a wound. We need to make sure wounds are covered while the skin heals to prevent infections. So how are our blood vessels adapted to their specific function? Red blood cells are very, very specialised cell. They do not possess a nucleus. They're the only cell that doesn't, and that means they have more space for hemoglobin. They're also a weird shape. If we turn them sideways, you can't really see it on this diagram, but they kind of look biconcaves. They dip in the middle. And I always think they look a little bit like a Yorkshire pudding, for example. And this biconcave shape means that they have a larger surface area, so oxygen can diffuse in and out of them really, really quickly. Inside is packed with hemoglobin, which attaches the oxygen to it, binds with oxygen, forming oxyhemoglobin in order to transport lots of oxygen from the lungs to the cells. And they're very, very flexible. Remember, capillaries are really, really tiny blood vessels, and these cells have to be able to squeeze into these narrow capillaries in order to get the oxygen to the tissues. White blood cells, it's the lymphocytes which produce our antibodies. They also produce antitoxins, which neutralize toxins that come from bacteria and other pathogens. The phagocytes can engulf and digest pathogens in a process called phagocytosis, which you learn about as well in the spec. And they have a large nucleus, but the nucleus is very flexible. And that means that these white blood cells can actually migrate from the blood into the tissue areas in order to get to pathogens infecting tissue. So they're very flexible in their shape. They can change their shape slightly to do that. And finally, the fragments of cells, the platelets, these don't have a nucleus because they're not really a true cell. But what they do is it's actually through quite a complex mechanism. You don't need to know about at GCSE, but they trigger blood clotting if a blood vessel is damaged. So they are very effective at doing that. We're going to move on in our last bit of the cardiovascular system topic area. We're going to look at diseases of the cardiovascular system. So the cardio meaning heart, vascular meaning vessels, and also how we treat them in this kind of current day and age. First of all, let's address, well, what is coronary heart disease, or CHD? This is an example of a non-communicable disease. That means it's not spread from person to person. It's a disease that someone develops in their lifetime, and they can't pass it on through spreading it, through sneezing, or even through genetics, generally speaking. Now, what happens in CHD, you can see this in my diagram, is that layers of fatty material start to build up inside the coronary artery walls. We can see this yellowy plaque forming. And if we do a cross section, that causes a narrowing of the lumen through which the blood is trying to flow. If the lumen gets narrowed, the artery gets narrowed, then the blood flow is reduced. And if this is happening in the coronary artery, that's the artery of the heart, the coronary arteries, I should say, because there's a few of them, then that's going to deprive the heart muscle itself of blood. And remember what blood is bringing. Blood is bringing oxygen. So if there's less oxygen being received by the heart muscle, then respiration rates are going to reduce, which means in turn less energy for the muscle contraction. And if the heart contractions are either weakened or even prevented, that's what causes a heart attack. The heart will go into spasm, or it will stop working completely. Now, there are things that can increase the risk of someone developing CHD. Risk factors don't cause it, they increase the likelihood of you developing it. What might they be? Smoking, high fat diet. We're talking about really bad fats here, like fats like lard and fats found in red meats, etc. Not exercising enough, just generally being overweight, high blood pressure that isn't addressed medically, high cholesterol. Now, there are actually some cholesterols which are good cholesterols. We're talking about the bad cholesterols here, but for GCSE, you can just call it cholesterol. And all those risk factors are going to increase your chance of developing coronary heart disease. Let me just skip back to that slide. I went too fast then. So it's really important that you are able to list some of those risk factors. They may ask you about that in an exam. Treatments for cardiovascular disease. Now, medical treatments are amazing these days at treating people who have this disease. Stents. Stents are a tube or a mesh tube which is inserted into a narrowed coronary artery, and then it's inserted into the artery. It's like a tube, and then it's opened up. And when it opens up the artery's lumen, so the blood can flow much more easily and maintain blood flow to the heart muscle. It's a really effective treatment for someone who's got a narrowed coronary artery problem, because this mesh tube, when it's inserted into the coronary artery, opens it up nice and wide, and it can last a long time. It's actually quite a simple procedure now, hospital-wise, short recovery time, and it can in fact remove the blockage that had already formed previously. However, there's always going to be disadvantages and risks. Having something inserted into a blood vessel around your heart is invasive. Risk of infection is high. It could damage the blood vessel and cause internal bleeding and even clotting where you don't want it. You have to have an anaesthetic to have this operation, and some people don't do well under anaesthetic. It can be a risk. And you would have to have anti-clotting drugs after surgery until you've healed. You don't want clots forming that then travel to your brain, because that could cause a stroke. But on the same kind of page, anti-clotting drugs means that you're prone to bleeding, which could be a problem if you had an accident and your body bleeds too much. So they are all risk factors. More treatments might include statins. Now, statins are drugs given to people who have high cholesterol and are at risk of becoming ill from CHD. What the statin does is it reduces the cholesterol in the body, and it also helps to slow down those fatty deposits in the walls of the arteries. So it's an amazing drug to help reduce the risk of blood clots, which can cause strokes and heart attacks, and it keeps your cholesterol levels at a healthy, stable level. But you have to take them for life. It's a long-term thing. Some people are rubbish at taking medication, they forget. And you can get side effects from statins over a long period of time. A lot of people complain of muscle pain, aches and pains in their joints, and liver damage. And it doesn't actually, if you've already got a blockage, it doesn't remove the blockage, unfortunately. It slows down any deposits which are forming, but if you've already got a blockage, it's not going to remove them. It's not like a miracle overnight treatment for a blocked artery. Other ways that we address heart problems involves how we can solve the problem of faulty heart valves. Remember that valves are found in between the atria and the ventricles. They're also found at the beginning of the pulmonary artery and aorta to prevent blood going backwards. Sometimes they're faulty, where they don't open fully, or they may leak, which means some of the blood does backflow, and it can then affect how efficient the heart is pumping blood out. It can affect a person's cardiac output. So let's have a look at an example. If the valve in the aorta is leaky, less blood is going to be pumped out of that left ventricle into the aorta, which means less blood in turn is going to move into the systemic circulation. Less oxygen reaches your body tissues, less aerobic respiration and energy. A person becomes tired and the muscle contractions become weaker. We're also going to get more anaerobic respiration happening in those areas where oxygen is not reaching properly, and that can lead to lactic acid buildup and muscle fatigue. Now, the reason I'm going through this example is in some of the kind of extended questions, especially in higher tier GCSE, they might ask you to suggest why a faulty valve can cause tiredness in a person or muscle contractions that are weaker. So you'd have to come up with a full scenario of events, like the story of what's happening that leads to that unfortunate symptom. Now, different valves exist. You can be given donor valves from humans. So obviously someone who's donated their heart after death, those valves can be transferred as long as they're still healthy to an individual who needs them. Some valves come from animals like pigs because they're very anatomically similar to us humans. Alternatively, you can have artificial or mechanical valves to replace faulty valves. So there's 3 possible places we can get valves from if we need them replacing: animals, humans, or man-made mechanical valves. Again, we need to evaluate what's good, what's bad, the pros and the cons. Again, another one of those longer questions you might get asked. Advantages to the mechanical valve is that they are much more long-lasting and durable, so they don't kind of get damaged very easily. And you're not going to risk transferring infection from an animal or another human into the person who's receiving them. But they do kind of encourage platelet kind of collection and therefore blood clotting. So people who are given mechanical valves will end up on anti-clotting medication for life to reduce the chance of that happening. You don't want a clot in your blood. It could travel to the brain and cause a stroke, or it could travel towards the heart and cause a heart attack. Biological valves, they're ones that come from living things like animals, like humans. Now, there isn't generally risks of blood clots around these valves because they're natural, but they don't last as long as the mechanical valve. They have a lifespan of about 10 years. Sometimes, if they're the wrong tissue type to your body rejects them, your immune system attacks them. And if we're using animal valves, there's quite a lot of ethical considerations where some people disagree with the use of animal tissue. It's not right. It's cruel, for example. And you could transfer pathogens or diseases from donor animals, donor humans, to the person receiving those valves. Heart failure then. So if you were unfortunate to have heart failure, but it was treatable, how might you be treated? A heart transplant. A heart transplant is when a heart is given to a person from a donor because the person's heart cannot pump blood effectively to sustain life. The pros, obviously, it leads to improved life expectancy and quality. And if it's a successful transplant, it should be a permanent thing. But again, if it's not that person's own tissue, there is a risk of the immune system rejecting it. When people have donor organs implanted, they are tissue typed, they are checked to make sure the tissues match as best as they can, but it's not always perfect. You also have to wait for a donor. You can't have a live donor in heart transplants. The person has to have died. So you're waiting around for that. Sounds a bit like depressing, but you do have to wait for an organ that's limited out there. Having an organ put into you from somewhere else also means that to reduce the risk of the immune system rejecting it, you would have to suppress your own immune system by taking drugs to do that, called the immunosuppressants. If your immune system is suppressed, then you're more at risk of getting other infections then. So it's a little bit of, pros and cons, swings and roundabouts. Artificial hearts. This is a bit of a weird one, but artificial hearts are temporary devices used in hospitals in extreme cases. Let's say someone has heart failure and they are dying, but there's no heart then at that moment in time readily available for transplant. So what that person would be hooked up to is an artificial heart to do the job of the heart while they wait, but it has to be done within a hospital and very, very, very closely monitored. So it's an immediate solution for critically ill patients. You don't have to wait for a donor. You can be kept alive basically. But again, high risk of infection and clotting because it goes into the body and it's mechanical, it's artificial material, it's not long term, it doesn't work as effectively as a normal heart, and you have to stay in hospital while you have this artificial heart inserted into your body. Overall, whether you're having artificial hearts or heart transplants, the big risk to both is that you are undergoing surgery. Surgery requires anaesthesia. Anaesthesia and anaesthetic is a risk for death. People can die from anaesthetics. And if you're opening up someone's body, you're exposing them to pathogens. So there's a risk of infection as well. The overview then of health and disease from what we've done so far. Health then. If we were to address the term health, what does it actually mean? Health is described as the state of complete physical and mental well-being. So it's not just about the body, it's also about the mind. If we were then to separate disease into categories, we can say that some are infectious or communicable and some are not infectious and they are non-communicable. And poor health can increase the risk of developing other conditions like infections or even mental illness. Remember that a non-communicable disease is one that we cannot transfer between organisms. It's not contagious, it's not infectious. What is a risk factor though? A risk factor is something that increases the probability of developing a disease. If you're exposed to a risk factor, it doesn't mean you're going to get that disease. It just means that you're increasing the chance of it happening. Not all people then exposed to risk factors will develop a disease. As I just said, it's to do with increasing the likelihood. Let's look at some examples. Types of risk factors and what disease that they're linked to, and we're talking non-communicable diseases here, the ones that you develop you can't spread. Smoking, the obvious one, is cancer. That can also give us problems with our cardiovascular disease, CVD, sorry. And obesity, too much sugar in our diet and too much fat in our diet is linked to diabetes and also heart disease. Alcohol is linked to liver diseases, brain damage, birth defects if you drink while you're pregnant. Poor diet and lack of exercise, again, cardiovascular diseases, type 2 diabetes. Genetics can increase our risks of cancers and other inherited disorders. If you have the genes for certain disorders, it doesn't necessarily mean you're going to develop them, but you could, it's a risk. Carcinogens are chemicals that cause cancer. So if you're exposed to a carcinogen like cigarette smoke, then that's going to increase your risk of lung cancer. Smoking or alcohol, especially during pregnancy, can increase the risk of the fetus developing problems with their own development. It can lead to deformities, for example. Let's focus then on that pregnancy bit and risk factors. Risk factors during pregnancy can impact how the fetus develops from an embryo. And that's because chemicals that we ingest can cross the placenta, and that can also impact as well oxygen delivery to the fetus. If you smoke when you're pregnant, the chemicals from smoke cigarette, carbon monoxide and nicotine pass across the placenta into the fetus's blood. The carbon monoxide will bind to hemoglobin on the fetus's red blood cells more readily than oxygen. And it means that the oxygen concentration of the fetus's blood is reduced. It deprives the fetus of oxygen. Nicotine, the drug on the other hand, is something that's going to narrow the blood vessels in the mother and the placenta and restrict blood flow, and therefore further restricting oxygen being received by the fetus. The consequences then, this can lead to premature birth because the fetus goes into stress if it's not getting the oxygen it needs. It can even cause miscarriage. It's where the baby dies in the uterus and then has to be born, unfortunately, dead. Low birth weight, where babies aren't getting enough oxygen, they're not having enough energy production from respiration, and therefore they're not growing adequately. And potential developmental delays, parts of the body not getting enough oxygen or not developing properly, and that can affect the baby after it's born in their lifetime, especially in the respiratory system. It can lead to respiratory problems in early life of the baby. Alcohol. Alcohol will freely move from mother's blood across the placenta into the baby's blood, and the baby hasn't developed fully yet, so unlike an adult, it doesn't have the right enzymes to deal with alcohol. It can't break it down properly, so the alcohol stays in the system of the fetus in very high concentrations for a long time, and we call this fetal alcohol syndrome, or FAS, and it's a very serious condition. What happens to the baby if it has FAS is it ends up with brain damage and learning difficulties. It can have facial deformities where its face doesn't develop properly, growth restriction. It can lead to behavioural and emotional problems, an increased risk again of premature birth and miscarriage. So there's lots and lots of effects of FAS, serious alcohol exposure of the fetus during pregnancy. Now, don't forget that risk factors also can interact. You can have multiple risk factors that can lead to a cascade of effects. We're talking like worst case scenarios here, but to be aware that if you do a lot of bad things, like you have a bad diet and you smoke and you don't do much exercise, then you are going to be exposed to multiple risk factors at once, and that's going to put you even more at risk of the consequences of that, like developing heart disease. Also, Socioeconomic status has an impact on people's well-being and health. That means, are you living in an area where you have good health access? What's your environment like? What's the risk like because of the impact of your social environment? There are parts of the world, for example, where disease is more rife because they don't have as good healthcare, as an example, or they don't have vaccination programs that reduce the spread of disease. So socioeconomic status is a slightly different risk factor. Okay, let's move on to cancer, another disease that the spec requires you to have some knowledge of. What is it? Cancer is uncontrolled cell division, mitosis gone wrong. It's caused due to a mutation, which means a change in the DNA of cells. So that's an important thing to remember. A mutation is the change in the DNA of a cell, which then leads to this uncontrolled cell division. And when those cells divide, they form a mass, and it's called a tumour. And these cells are abnormal. They're not normal cells, and they just keep dividing uncontrollably. But tumours come in two different types. We have benign and we have malignant tumours. A benign tumour is when you have abnormal cell division and a growth, a lump, but it doesn't spread, it just stays in one part of the body, and it can be removed, and quite often very successfully. The only problem with benign tumours, if, for example, if they're deeply embedded in an organ, and to get to them to cut them out, it could damage the organ. A malignant tumour is more the cancerous tumour. What that means is they can metastasize, which is this word here, metastasis, I can never say it, metastasize, which means they can spread. What happens is the cells of the tumour break away from the main tumour, the primary tumour, enter the bloodstream, and then travel to other parts of the body and form secondary tumours elsewhere. Metastasis is the term to describe that process. Malignant is another word to describe that a tumour is spreading to other parts of the body. Now, malignant tumours are definitely considered more dangerous, although I mentioned before that benign tumours can be problematic if they're deeply embedded in an organ, affecting the organ's function, difficult to cut out. Generally, malignant tumours, because they spread and form more tumours around the body, they are deemed by doctors much more difficult to control and get rid of, and also much more dangerous. So what can cause cancer? Carcinogens. Carcinogens, we mentioned before, are chemicals or substances that are known to cause abnormal cell division. Typically, tar in cigarettes and alcohol are carcinogenic agents. So if you're exposed to a lot of them over your lifetime, you put yourself at risk. Risk factors for developing cancer: your environment, your lifestyle, smoking. Are you overweight? Are you exposing yourself to the UV light of the sun? Are you drinking a lot of alcohol? All of these things are risk factors increasing the likelihood of you developing cancer. But genetics does play a part in cancer as well, unfortunately. There are some genes within families which can lead to tumour development, and they are faulty genes. The ones that are most common are genes that cause some kinds of breast cancers and some kinds of ovarian cancers in women, and they can be passed on from parents to offspring, unfortunately. How do we treat these cancers? Now, it very much depends on where the cancer is and what type of cancer it is. But what a doctor will have to determine first of all is the type of cancer, where it is, how big it is, and whether it's spread. So if someone goes to the doctor with a lump, the lump will be investigated. They will have a biopsy where cells from the lump are removed. They will check it to see if the cancer is benign or malignant. They will measure the tumour with a scan, and they will also scan the body to look for cancer spreading elsewhere. Once they've determined what the type of cancer is, then they will decide what to do about it. One of the first things they will do is try and remove the tumour from the body. using the surgery. Now, this is more common with benign tumours because they haven't spread, they're localised. We don't tend to always do this with cancer that has already spread in the body. Radiotherapy is when the cells that are cancerous are targeted specifically with radiation. And what it does is the radiation damages the DNA of the faulty cells, and the cells can't divide, and it prevents the tumour growing. So it literally, you'll go for radiotherapy treatment, they will bombard that area of the body with radiation to kill the cancer as best they can at the tumour site. Unfortunately, it can also damage cells around the tumour and can cause people to have areas where that tumour was being very sore and irritated. It can even cause hair loss in that area where the radiation is targeted. Chemotherapy is used if a cancer has spread. What will happen is a person will be infused with a drug, so it will be like a liquid form of a drug, through a drip into the person's blood system, into their circulation, and that fluid with the chemicals in the chemotherapy, which are chemicals to kill the cancer, will then go right through the circulatory system and reach all parts of the body and kill cancer cells throughout the body. So it is used for metastatic cancers that have spread. However, these anti-cancer drugs are so effective that they also kill healthy cells that are naturally fast dividing cells, because fast division is a typical sign of a cancer cell, but we also have healthy cells that naturally fast divide, like your hair follicles and the cells that line your digestive tract. And therefore they get killed as well. So people lose their hair when they have chemo. They can also be very sore inside. It makes them very sick. It gives them nausea. And it also kills off their white blood cells, which gives them immunosuppression. So they're very, very susceptible to infections. Just to then go back to our key terms, things that you really need to make sure you understand the meaning of. What is a tumour? It's a mass of abnormal cells that divide uncontrollably. A benign tumour is one that does not spread, it's non-cancerous, and a malignant tumour is a cancerous tumour that can spread from the primary tumour to form secondary tumours throughout the body. Moving on now to our plant tissues and how substances are transported in the plant. We're going to start off by looking at the leaf. Now, the leaf has numerous layers which are all extremely important for either absorption of light or for gas exchange for photosynthesis. The leaf itself is an organ. Because it's made of lots of different specialised tissues, that's what puts it into the organ category. And its function, its main function, is photosynthesis, as I said. Gas exchange is not just about photosynthesis, that can also be about respiration, and we need a transport system bringing substances into and out of the leaf as and when they are needed. We'll have a quick look at the different tissue layers. So you'll see on the picture at the side of the slide, we have the top of the leaf here, and then we're going to look down the leaf at how the tissue layers change. First thing to focus on is our upper, or our cuticle. Our cuticle is basically a waxy layer. It's transparent, it allows the light to pass through, but it also offers a degree of protection from too much water loss by vaporization, or evaporation, I should say. As I said, it is transparent. We need the light to pass through it, because when the light passes through it, it has to then pass through another layer, the upper epidermis, and then into the palisade layer. The upper epidermis, also transparent, also acts as a protective layer. It can actually secrete some of that waxy cuticle to form the waxy cuticle on the upper surface of the leaf. The palisade layer, this is the main tissue area of the leaf that has photosynthesis happening at a very fast rate. These cells are packed with chloroplasts, which means as the light hits them, you're going to get a lot of absorption at this point, which provides the energy for the reaction. If we look at the tissue layer under that, we then have this spongy layer. Now, the spongy layer consists of cells which are not as tightly packed together. They are loosely packed together, which means there's going to be lots of air spaces where gases can easily move around inside the leaf in order to enter and leave the various cells for photosynthesis or respiration. You'll notice as well that within that kind of tissue layer there is another tissue called the vein or the vascular bundle. The vascular bundle is made up of two types of vessel, the xylem and the phloem. The xylem is the vessel that moves water and mineral ions upwardly through the plant from roots right up to the leaves. The phloem is another tube, another vessel, but it has a different role. It actually moves sugars that are made through photosynthesis to all different areas of the plant, either to be used or to be stored. And then the last section of the leaf is our lower epidermis. Now our lower epidermis is pierced with stomata, so we see lots of these regular kind of pores throughout this layer, and the stomata are there to allow gases to move in and out, but they also are an exit point for water vapour, which drives water movement through the plant but can also be detrimental to the plant if too much water is lost on a very hot day, which is why there are also guard cells present in this lower epidermis, and those guard cells can change shape, which we'll mention in a minute, in order to open and close those stomata and have some degree of control over water vapour as a loss of water from the plant. So here we have the kind of picture again, but just much bigger, so you can really see these different layers. It's really important in GCSE biology that you can identify these layers and label them, and then briefly describe their role in terms of photosynthesis and gas exchange. Let's have a close look then at the guard cells and the stomata. Now, the guard cells, as I said before, can change shape. They have to be able to change shape in order for the stomata to either be open or closed. Generally speaking, in a normal environmental situation where conditions are kind of stable, plants will have open stomata during the daylight so that photosynthesis can happen, because they need carbon dioxide to enter the leaf and they need oxygen to leave the leaf as well. The guard cells will therefore be in the right form, the right shape in order to open that pore. They're a bit of a weird shape, the guard cells. They're like irregularly shaped cells. They're kind of curved shape, like so. And these two guard cells, when they sit next to each other, because they're of the shape that they form, they create the hole in the middle. It's literally a space in the middle that we call the stomata. At night, the guard cells change shape in order to close the stomata off, and that helps to prevent too much water loss. And also at night, they're not going to have that need for carbon dioxide, because photosynthesis can't happen at night because there is no sunlight. So just to reiterate, the stomata are the point of gas exchange, carbon dioxide in and oxygen out, but water vapour can also leave the leaf when the stomata are open. And when water vapour is lost from the leaf surface, we call this process transpiration. So it's the evaporation of water from the leaf surface. Carbon dioxide is the gas needed for photosynthesis, remember, and oxygen is a product of photosynthesis. Now, oxygen is useful to the plant because the plant can actually use it for its own respiration, but a lot of the oxygen it doesn't use will end up leaving the leaf through the stomata into the atmosphere so that other organisms can use that oxygen for their respiration. Bit of a maths application now. Now and again you might be given a question which requires you to calculate the number of stomata in a field of view. So here's our question. Student counts 15 stomata in a field of view. This is a square field of view, not a round one as you quite often see under the microscope. Your job is to estimate the number of stomata per millimetre squared. First thing you'd need to do is find the number of stomata in the picture. In fact, they've told you that, haven't they? So they've told you there's 15 stomata. So we can see here we've got the stomata, these little holes here, and then these surrounding bits there are the guard cells. There's your guard cells that I'm just shading in red. And the bit in the middle, the gap in the middle, is the stomata. Now you already know that there's 15 stomata in this field of view, but what you have to do next is work out the area of the field of view that you're looking at. Now you're given information here to calculate that. So we've got 0.5 millimetre that way, 0.5 millimetre that way on the picture, on the image. So you'd simply use your normal working out an area calculation, 0.5 times by 0.5. So the area of the field of view is 0.25 millimetre squared. Don't forget that we've worked it out as an area. You will then, just get rid of that for a second, you would then need to account for how many therefore would there be in 1 millimetre squared, because that's what the question wants you to do. So you'd simply take 15, because that's the number of stomata, divide it by the area you're looking at, and that should, once you've plugged it into your calculator, come out at 60 stomata per millimetre squared. Transport then. We're going to have a little look at how the xylem is adapted for its role in water and mineral ion transport. Now the plant's transport system runs right through it, from roots into the stem into the leaves, and there's many things that need to move through that system: water, mineral ions, and also sugars. Now in terms of the xylem's role is to move water and mineral ions up the xylem. It always moves upwards. Now in order for the xylem to do this really well, it's very adapted to its function in its structure. Water will always move upwards in one direction only, and that's driven by the transpiration that happens at the leaf surface, which we'll take a look at in a minute. But before we do, we're going to have a quick focus on how the xylem forms as this tube. Now initially, when the xylem is forming, it starts with cells, normal plant cells, that lie in a longitudinal fashion on top of one another as they grow. So they grow in this like longitudinal form. But then what happens is these end walls here, here, and here, and here, and here on my diagram, they get broken down. And as a consequence, the contents inside all of those cells literally falls away and leaves a tube-like structure that's empty. So effectively, the xylem is a dead tissue, but it doesn't decay, and it also becomes extremely strong. And that's because as it develops, a special chemical called lignin gets laid down in the walls of the tube. You can see it on this diagram here, these little squiggles. And that is a really, really strong waterproof chemical that doesn't decay easily. So it makes this perfect tube, really strong waterproof tube with nothing in the way for water to be able to just be moved upwards. If you were to describe, therefore, the xylem's structure, you would describe it as being dead and hollow, and it's joined end to end. The cells are joined end to end to form these long tubes. Lignin is a really important chemical to remember that gives the xylem its rigidity and its waterproofness, if that's what we're going to call it. So it's a very rigid, that means strong, difficult to break down, difficult to bend structure. And the end walls, remember, disappear during the xylem's development. So there are no end walls, there are no contents in the xylem, which means there's nothing in the way for water to flow. It's not impeded by anything. There's no organelles in there either. Remember, they've all fallen away. All the contents of the xylem have fallen away. Nothing is restricting water's flow. So in terms of water moving upwards, we describe the whole water movement up the xylem as the transpiration stream. We'll take a closer look at that now. The transpiration stream is the movement of water with dissolved mineral ions in the water up a plant from its roots into the stem and then into the leaves. It's a passive process. Passive meaning it's not driven by energy from respiration. It's actually driven by the energy of the sunlight, the heat energy from the sun. How does it work? We always start with the leaf because this is what's driving the movement upwards of the water through the plant. When the stomata are open, so this is going to be in the day, water will have a tendency to evaporate through the stomata of the leaf into the atmosphere, and that just process at the leaf surface is known as transpiration. Transpiration is the evaporation of water from the leaf surface. When that water is lost, you can see it here in the diagram, that is then replaced, that water that's lost in the leaf is replaced by more water from the xylem tissue in the leaf. And in turn, that pulls water from the xylem in the stem. So this pulling of water up the xylem and through the leaf happens because of evaporation of the water through the leaf surface. At the same time, water is continuously entering the plant at the root. We can see here that this is our root area, is our root hair cell. Water will be coming into the roots via the process of osmosis to replace the water lost. So as that comes in, it'll be pulled upwards, pulled across the leaf, and then out through the stomata. This whole movement right through the plant, from the roots up the stem and out through the leaves, is called the transpiration stream. Just to go back to the stomata again, because these are vital in this process as well. They're obviously there for gas exchange, but the stomata have to also be open for this water vapour to exit, which in turn will pull the water up the plant and help with the transpiration stream. The guard cells can open and close the stomata. There will be times where the plant doesn't want to have its stomata open because it's at risk of too much water loss. That would typically be in a very, very hot environmental conditions in the middle of a hot summer's day, for example. But most of the time, in normal bright light conditions, and as long as there's lots of water available, then the stomata will be nice and wide open and allowing for carbon dioxide to enter the plant for photosynthesis. In the darkness or in the drought, the stomata will be closed down to reduce the chance of too much water loss through the leaf surface. And the way the guard cells work is when they open the stomata, it's because they are what we call turgid, which means they're plump and full of water. And when they're plump and full of water, they form this kind of curved shape, and that creates the stomatal aperture in the middle. That's our stomata there. And in the darkness, the guard cells become flaccid, they've lost water, so they literally kind of deflate a bit, and as a consequence, they kind of collapse on each other, and the stomatal hole in the middle, the pore in the middle, closes down. Transpiration then is the loss of water through the leaf surface, through the stomata, driven by evaporation of water from the mesophyll cells of the leaf, and then it diffuses through those stomata into the atmosphere. So if you were asked to just define what transpiration is, that is your kind of perfect model answer to write down as your exam answer. Don't forget that water is continuously pulled up via the transpiration stream in the xylem, and that is the whole process of water movement. Transpiration is just the evaporation of the leaf surface. The transpiration stream is the entire movement of water from the root up the xylem of the stem and then across into the leaf and out of the leaf. All sorts of factors can have an impact on the rate of transpiration. That's how fast it's happening. And most of them are obvious, but it's important that you're able to explain biologically why these factors have an effect. We'll have a quick look at these then. So temperature, if there's a higher temperature, transpiration rates are going to be faster because there's more evaporation of water from the leaf surface. Humidity, now you can look at this either way, but just so you're aware, humidity is how much moisture is in the air. The lower the humidity, the drier the air, the greater the rate of transpiration. Remember that dry air would create a really steep water potential gradient or water vapour concentration gradient between the inside and the outside of the leaf, and that means water is going to diffuse more efficiently, more effectively, faster out of the leaf on a very dry day, and it would be the opposite if it was a humid day. Air movement, we're meaning wind here, wind speeds. If there's more wind, transpiration rates will go up, because as the water vapour moves out of the leaf, it is then swept away by the wind and then is replaced by air that's depleted water vapour. So it maintains that water vapour concentration gradient as well. And the light intensity, that's because stomata open generally in the light. They're nice and wide open, and that means more photosynthesis can happen, but also you will get more water loss and more transpiration. Opening and closing of the stomata. We've kind of covered this already, so this is just to sum it up. Stomata open in the light for carbon dioxide to enter the leaf for photosynthesis, and it's the guard cells controlling the opening and the closing of the stomata. When the water moves into the guard cells, that would be by osmosis, because it's water crossing a membrane to enter a cell. Remember, the cells become very turgid. We talked about this a minute ago. They bend outwardly, and that's what creates the stomata in the middle. And at night, when there's less photosynthesis, the guard cells will close the stomata to conserve water, and that's because they are more flaccid and they become a little bit less plump and they don't bend as much. In terms of how we would measure the rate of transpiration, the equipment that we'd use to do this is called the potometer, a bit of an old-fashioned piece of equipment. What it consists of is this glass tubing which is attached to a capillary tube that has a ruler on it, so you can measure a distance, and there'd also be an area where you could put your plant to be investigated and a beaker of water. How does it work? Now, the potometer allows us to investigate a variety of things about transpiration. We can just look at the rate of transpiration from our plant generally through this piece of equipment, or we can compare plants and their transpiration rates, or even we can set up different environmental conditions around a plant to investigate them. So a humid day, for example, we could mimic a very light day, a windy day, in a variety of different ways. The idea of the potometer, though, is it's very much an estimate of the rate of transpiration, because water moving through a plant isn't all lost through transpiration. Some of that water will end up going into cells and being used by the cells for various things. So just to be aware that this is very much just an estimate, a piece of equipment used to estimate the rate of transpiration from a plant. Now, when you set up the potometer, you have to ensure it's absolutely airtight. We don't want air bubbles entering the plant xylem, because that breaks the water flow and transpiration would come to a halt. So it's quite hard to set up. You have to do it underwater, and you'd have to cut the shoot you're investigating from its main plant underwater as well, to try and prevent any air bubbles entering the equipment at all. So effectively, you're filling up this equipment with water and you're putting your plant into the equipment underwater. Now, you'll notice there is an air bubble here. That's because we purposely then introduce one air bubble into the capillary tube, because that air bubble is what we're going to follow in order to estimate our rate of transpiration. How do we do it? We would need to get a clock, a stop clock, because we want to time it. We would let the plant kind of adjust to its environment, or whatever conditions you've decided to put it in for your experiment. To introduce the bubble, you would simply get your beaker of water here and move it away for about a second from the capillary tube and then put it back again. And as you do that, an air bubble gets sucked into the capillary tube. So here's our air bubble we've introduced. And you generally then time the air bubble's movement and then measure how much the air bubble moves. So you'd literally say the air bubble moved that amount over this period of time, and that enables you to get an estimate of the rate of transpiration. The distance the bubble moves over a period of time is what you're trying to work out here in order to get some results for your investigation. So things you could investigate using a potometer: you could investigate whether light intensity has an effect on the rate of transpiration, how quickly that bubble moves. How would you do it? You could use a lamp. bench lamp and do it at different distances from your plant and then repeat the process over and over again. You could mimic a windy day by putting a fan next to your plant at different speeds and see if that affects how fast the bubble moves along the capillary tube. Or you could investigate different temperatures by setting up a heater and adjusting, obviously, the temperature the heater is at. And the humidity could be changed. You can literally put a see-through bag around your plant, but spray water into it first to create humidity in the atmosphere around the plant. Just going back to the previous slide for a second. So let's say you were setting up a humid day. You've got a plastic bag around your plant. You've introduced some water spray so it's nice and moist inside. What will then happen is as evaporation of water from the leaf surface takes place, or transpiration, that means that water is going to be pulled up the xylem from this equipment, which in turn pulls water along the capillary tube, which means the bubble is going to be pulled along too. So the bubble will be pulled along as the water moves up and out of the plant, and you're simply going to measure how far the bubble moves over a period of time. The reservoir is there to reset the equipment. So what you might do is you might say to yourself, right, I want to repeat the experiment to see if I get the same result. You want to push the bubble back again to the start. So you open this little tap on the reservoir and the water flows down and it pushes the bubble back and you can start your experiment again. In this experiment, you also need to consider your dependent variable on exactly what that would be and the units you would apply. Remember, you're measuring the distance the bubble moves in a time period, so you're going to work it out in, for example, millimeters per minute or millimeters per second, depending upon your choice of time unit. And don't forget that there are lots and lots of control variables. Now, these would differ depending upon what you were investigating. So I'm just going to take, for example, if you were investigating light intensity, the light intensity would be changed as you do your investigation over and over again with the photometer. But you would want to keep the temperature around the plant the same throughout and for every repeat. So you know that the temperature isn't going to be having an effect. It's only going to be your light intensity. And you also want to make sure that you're using the same plant throughout as well, because different plants might have different surface areas on their leaves and therefore different numbers of stomata. So there's lots of control variables to be considered, but it depends upon what you're investigating. So you'd have to think carefully about that if you were given a question. Make sure you know what the dependent variables are, what the independent variables are or is, and what the control variables would be. So our final part of our plant tissues and plant transport is focusing on the other vessel found in the vein or the vascular bundle known as the phloem tissue. The phloem is found next to the xylem tissues. They're in the tubes literally in bundles that run right through the root, right up the stem and right into the leaves. But the phloem has a very different function and a very different structure. What actually is it then? It is a transport tissue, but what it transports are the dissolved sugars that have originally come from photosynthesis. Photosynthesis gives rise to glucose, but glucose doesn't get transported through the plant because it's too soluble. It gets converted into a less soluble sugar called sucrose, and that sucrose then is moved to other parts of the plant. But why? It might need to go to growing regions which are carrying out a lot of respiration. So moving that sugar to those areas means that it can be used immediately in respiration, providing energy for growth to happen. Or the sugar might be stored, so it might not be needed straight away. Therefore, the phloem would transport that sugar to a storage organ. Roots typically, tubers are kind of like root vegetables are tubers like potatoes and things like that. And there, that sucrose will be converted into a storage carbohydrate called starch and can be used at a later date should the plant need to convert it back to sugar. The process by which the sugar moves is known as translocation, so it's got its own unique name as well. Trans means sort of to move different places, so trans to transfer, location, the place as to where the sugar was. So it's moving it around the plant, essentially. Moving it through the phloem, as we said, it's always phloem that moves the sugars. The xylem moves the water, remember, up, and the phloem moves the sugars. But ironically or differently to the xylem, the phloem is able to move the sucrose both upwards and downwards. And we also have come to understand that this requires energy from respiration to happen. It's therefore an active process. It's not passive like transpiration is. Translocation is really, really important. Really important. We said this a little bit earlier because the sugar in the leaves that's made during photosynthesis might need to be used in other areas of the plant that can't photosynthesize, or it might need to be stored for a part of the year where the plant isn't able to photosynthesize enough, like the winter. So by having a storage of that carbohydrate as starch in the roots, for example, would provide the plant with sugar for respiration in the winter to keep it alive through those winter months. A bit about its structure then. Just like the xylem, the phloem starts as tubes that form from cells that grow in a longitudinal fashion. They grow on top of one another in a longitudinal way. But unlike the xylem, it's not dead tissue because the tubes still have in them cellular components and cytoplasm, and there's a reason for that. When the cells form the tube, what happens is the end cell walls don't completely disintegrate like they do in the xylem. They just perforate, which means little holes form in them. And the holes that form are not enough for the entire contents of the cells to disappear or fall away, but it's enough for a little bit of the cytoplasm and substances in it to come away, leaving space for sugars to travel. Now the little ends where these perforations, these little holes have happened, we call these the sieve plates. So these little pores, allowing sugars to move up and down the phloem tissue effectively. Now, because we know that the phloem can move sugar up and down and we know it's active, we have also come to realise that the phloem actually gets help to do this from nearby cells called the companion cells. Companion cells, you can see on my diagram here, lie next to the phloem tubes, and they are full of mitochondria, meaning that they are respiring at a very high rate and providing a lot of energy, which is then used for this translocation, this movement of sugars to different places of the plant that is happening in the phloem tissue.
If you found that video useful, then don't forget to hit subscribe, because we are going to be putting loads of videos out there on GCSE content leading up to your exams in the summer. And if you click the link below, you can sign up to one of our courses where you'll get access to 2 live lessons every week, workbooks that go with the lessons, plus homeworks, plus video solutions with all the answers to those homeworks. So hit subscribe and get signed up to one of our courses.